good morning, and welcome all you enthusiasts for invertebrate zoology, morphology, and evolution. I see there's a wide range of Disney sidekicks that are capturing your, if you look at them and immediately think back to watching them on screen, which one of them represents you most today. I put in the chat, mine is Ray, the firefly from Princess and the Frog, because he is, if I remember the film correctly, and it's often very, it goes in ebbs and flows, but it has been very popular in our house with our boys. My memory of Ray is that he is enthusiastic for a lot of things, and he gets battered all over the place. And that's kind of how I feel about this past year. I have enthusiasm for a lot of things, and this year has battered us all over the place. All of us. So, let's start with congratulations. Put your hands up in the air, pat yourself on the back. You're here, and you're doing fine. So, well done you. Okie doke. I'm going to save that and clear it and hide that. And we are going to continue. I will keep working the door. Hello to everyone who has just arrived. Today we're going to be talking about things neem. There's a phylogenetic relationship between the nematodes and the Nematophyrens that we'll be talking about. Those are ecdysozoans. But we're going to start out by talking about the Nematerens, which is our last spiralian group. And I'm going to even start off with something different. And that is the fact that you might not know this already, but since you have met, in lab, you had the opportunity to meet Dr. Adam Brunk, former student at the University of Guelph, who is currently a taxonomist of staphylinids, of rove beetles at the Canadian National Collection of Insects, Arachnids, and Nematodes that we're talking about today. This is Taxonomist Appreciation Day. Hooray! Why might you, and I like the wordplay, taxonomy, the study of organisms and how you phylum. You get it? I got it. It appeals to me. Taxonomist Appreciation Day is something that we should be doing as biologists, whatever stripe of biologist. If you're going to become a strict, straight-up physiologist, through to a conservation biologist, through to a sociologist who works with First Nations and Indigenous peoples, all branches of biology kind of intersect with taxonomy. This is an infographic from an executive summary of the state of Canada's taxonomic resources, or perhaps more correctly, a state of Canada's taxonomic crisis from a few years ago. You can see in the infographic that we're talking about the current gaps, that there's many, many gaps. There's an absence of national collection standards. There are many of the places that they're housed are inadequate and have no capacity for growth. Many of the highly qualified personnel that have worked on these groups are retiring and not being replaced. Traditional community knowledge is at risk, and there's an enormous number of consequences there that go from medical to esoteric to economic. And so, for example, we can't identify the next arriving zebra mussel if we don't have a taxonomist who specializes, who can give us the capacity to identify the next arriving zebra mussel, particularly in different life history stages. And so, as you think about positive thoughts about the taxonomists in your lives today, make sure that you appreciate them and think positive thoughts about taxonomists on today, Taxonomist Appreciation Day. So, like I said, today is things nema, which is Greek for thread. That actually describes the nematodes and the nematodes and the Nematophyrens, but it doesn't describe the nem with an E. Terens, which is where we're going to start. These are three different phyla that we're probably going to get to today. There might be a chance that I will carve off one of them for a little asynchronous quick blurb, because there is a lot to cover. There's a lot to cover in terms of morphological strategies, diversity, known diversity, unknown diversity. There are a lot of crazy things in the things named neem. So, to start out, Chapter 12 talks about our last spiralian group, the characteristics of the Nematerens. So these are marine groups, marine, freshwater, terrestrial, but principally marine. They're coelomate, they're triploblasts, so they're like the rest, many of the rest of the spiralian groups that we've talked about. They do have a complete gut, but then look at a couple of the next of those, point number 4. So complete gut, digestive tract complete with an anus. That's a coelomate. That's a characteristic of an organism with a body cavity. But they have their excretory system is characterized as with protonephridia. That's unlike those. That's more like a platyhelminth flatworm than it is like any of the other large and body cavity containing, like the annelids and the mollusks, spiralian. They have a super gorgeous, unique proboscis situated in the front of the body, and I've tried to gather some video of that to show it to you. A closed circulatory system, so that might lead you to suggest or imagine some things about their life history. If you think back to what we talked about with the mollusks, where we had an association between an open circulatory system and a more sedentary life, and a closed circulatory system with a more active life. Gonochoristic, but they can reproduce asexually. Keep letting people in. Hello, hello, hello. So these ribbon worms are mostly ambush predators. Think back to what we just said about the closed circulatory system. They have an aversible proboscis, the Wrinkle Seal, through gut, closed circulatory system. There, a summary of the previous summary. Some of them are gorgeous, and sometimes gorgeous, highly colored things, you can make draw a rough correlation between the more colorful it is in wavelengths that we can perceive, the greater the likelihood is that we have named it, or we've hypothesized about who could be named this. This generalization doesn't actually hold here. This quote at the bottom from Sunderberg in 2010 in a paper in Molecular Ecology talked about the fact that even though there's roughly 1,300 named and described species, almost half of those were described before 1900. And some of those descriptions, which you've had the chance to explore species descriptions, many of those written in before the start of the last century are so vague and incomplete that we have no particularly clear idea what it is that they're referring to. So this is a place where it's good to be talking about Taxonomist Appreciation Day on the Nemertean roundworm or the ribbonworm day, because we have two different problems here. We have the problems where we have 1,300 names, but we don't know how to apply maybe half of them. And I'm going to unpack for you that we have the problem, as we've seen with a lot of other groups, where we have unappreciated diversity. Diversity that morphologically doesn't make itself so clear to us. We don't see it. But they apparently do, because when we look at genetic differentiation amongst these morphologically very similar things, we see deep, deep genetic divisions that suggest to us that these are, in fact, separate species, otherwise morphologically cryptic. Now, let's take a look at this. So what you're looking at is an annelid on the bottom. You can see it's the parapodia, but what you're looking at top with here, with that kind of flipped open lid, what you're seeing is the death of the annelid as it crawls or is drawn into the start of that complete digestive tract of this long ribbon worm. So this is another look at one of these ribbon worms. This is, and the apomorphies or synapomorphy for this group is this Wrinkle Seal. So the trunk like this coelom, and this is a fluid-filled chamber in which the proboscis, or I suppose in this case the trunk, lies. Cerebratalis, this is a genus of ribbon worms there that is going to appear later on in one of the narrative stories about how little we know that we know we know. One of the other synapomorphies for the group is the loss of Chidae. You're familiar with this, but that's yet another example where a defining kind of apomorphy for a larger, more inclusive clade is lost in one of the daughter clades. What does that Wrinkle Seal look like? Well, here's a look at a terrestrial one. This is, you're just looking at the body right now until you poke it.
Oh, my goodness. Have you ever seen one of these?
I love videos from Australia.
You ready?
Stop. I love these videos from Australia. So there you saw the Wrinkle Seal get everted. Boom, there, with the delight of the Australian holding it. So let's zoom in and take a look at the body wall of one of these. Keep letting people in, and the body wall of what these look like. So we have this Wrinkle Seal. So this is this interior cavity where there is a proboscis and a sheath, and that can then be everted and used to predate. Remember, they're mostly mobile predators. They have cephalic glands that are secrete slime that facilitate moving and burrowing. The body, as you can see this in the label, the mesenchyme there, the body is largely muscle. So these are we've got layers of longitudinal and circular muscle. This is our packed, kind of a dense. This would not be a floppy thing if you were holding it. You would feel the density of that animal in your hands. So circular and longitudinal muscle, and then the mesenchyme. This is, as kind of a fact to wow your friends at trivia night, this is one of the uncommon examples where we see circular muscle inside longitudinal muscle. This is a slide of a cross section. So we've got the epidermis and the dermis, and then circular muscle contained or sandwiched in between longitudinal muscles here. So more about the Wrinkle Seal. This is this fluid-filled coelomic cavity. You can see it here and then squeezed out. And there's 3 different examples of species there. There's our delighted Australian, or another, actually this is another one, isn't it? Yeah, so this is another person, another hand, let's presume it's an Australian, and there's a delighted Australian accent there, as that Wrinkle Seal gets everted. And you can see, unlike the last one, this is a much more complicated kind of medusoid-like. Now, they can be tipped with these stylets, these calcareous barbs, that aren't known to be directly venomous, but they do secrete neurotoxins and other adhesives. There's the stylet, there's the Wrinkle Seal. Now, this is a video, you can go and watch this later on YouTube if you'd like. I think I sped this up, or I slowed it down, one of those. So you can see the proboscis getting whipped out, hitting the nematode, and then this hood opening up. I'm sorry. Keep my hands off the play button. And then perhaps the world's scariest Muppet or puppet, and boom, boom. And I'm sorry, I said that was a nematode he was eating, but maybe as you watched the video, you could see there were segments in it. So that's an annelid. That's a worm, a true worm. So carnivores, they can be armed or unarmed, referring to that stylet and that stylet bulb in the front part of the proboscis. They have this through gut that characterizes. So we have the stylet. And then if we take a look at their internal transport, you can see blood vessels draped around the Wrinkle Seal. They have no gills. There's a coelomic circulation via ciliary beading. So again, this kind of, the ribbon worms seem to have an intersection or a combination of traits that characterize truly coelomate and protocoelomate or pre-coelomate animals. Asexual reproduction by fragmentation, so you can, as this elegant drawing from Rupert, Fox, and Barnes shows, you can cut them into pieces, and then eventually you see them assuming full capacity later on. External fertilization with these egg capsules extruded into the external environment. And then indirect and direct development. So there is a trochophore stage, so that characterizes many of the spiralian groups, but they also have a new kind of larval stage that is called the Pylidium. And so we see these invaginations here of the ectoderm that characterize the Pylidium larva.
This is the iconic Nematode larva called the Pylidium. It looks like a hat
gorgeous.
with ear flaps. The ear flaps are called the lappets. There's also these two lobes. You can see a ciliated band spanning the lobes and lappets, an apical tuft at the anterior end, and inside the Pylidium you can see its gut, which is this dark brown area.
I love that. I think if I was going to come back in another professional capacity, and I'm imagining for a chance that I was designing spaceships associated with science fiction films, I think I would just try and merge everything I know about marine larval stages and just make as many spaceships for as many science fiction films as I could that look like different larval stages, like a villager larva spaceship, or a trochophore larva spaceship, or a Pylidium larva spaceship. I don't know if it would leave me very professionally satisfied, but it would lead me to be very personally satisfied. So this is one of the reviews that Per Sundberg, this is another of our, this is a somewhat optimistic Swedish taxonomist who deals with ribbon worms. And this is, I talked about a quote of his earlier that talked about everything for about half of the 1,300 names was not useful because the descriptions are so poor that you don't actually know what they're talking about. This is a more recent publication where he talked about degrees to which it is still, the systematics of this group are still problematic because many of them are not referring to monophyletic groups. And this is one case, in fact. So we see this is the genus Ceratobolus. These are barcode sequences. They are DNA sequences, mitochondrial DNA sequences for nominally things, multiple species. But if you look if you're me, if you take off your glasses and look at that computer screen very, very closely, what you'll see is that there's a whole bunch of taxa that have the same name that appear in different parts of that tree. This is really suggestive of the fact that those are, in fact, not the same species at all. If you take a look right up at the top here of this phylogeny, you'll see that the scale bar is 2%. So every bit of that distance down here, 2, 4, 6, 8, 10, 12, 14, these are like in between this group of 1 and this group of 1 on the outside, that we're talking like 20+% sequence divergence. So not at all what you would expect to fall within the same species. But if you look between these samples here in red, you can see it's really unlikely that these morphologically very similar things are in fact the same things. So this is one of the better known genera of the Nematerens, and here, even here, we find evidence that they are hiding cryptic diversity, that we are unaware of the diversity of the way that they see themselves in the world. And this was actually, maybe this came across your news feed late or early last fall. This is a news release about a particular study where there was a new Nemateren species found in Panama, and the first one for its genus that was found in the Caribbean, and it was found basically on the doorstep of the Smithsonian Tropical Research Institute, which is in Panama, along the canal. And as an aside, one of the director for the marine portion of the Smithsonian Tropical Research Institute, who is quoted in this article here, she is giving next week's virtual departmental seminar that is on this Zoom address next Thursday at 11:30. If you're interested, you can watch the departmental website for links and for announcements about that. But you can see here, again in the bottom, this is the newly described species just highlighted in yellow amongst several of the other taxa that they captured. So you can see clear evidence that there's a lot of things that look fairly similar in this group that we are only now appreciating that they are very, very divergent. And so I guess what I'd like to do now is take a second, that because it's Taxonomist Appreciation Day, and we've talked now about the Nematerens for a few minutes, about the role, the advantages that DNA played in those descriptions. And so what I'd love you to do is jump into the chat for a second and Tell me about what you think should happen to the morphologically depauperate groups like Nematerens and many of the other vermiform groups that we've dealt with. How should we be dealing with these? What advice would you give taxonomists about how to deal with the morphologically depauperate things? So I'm going to jump over to the chat for a second, see if I've missed anything. Yes. How big would the Pylidium larva be? Well, very, very small millimeters we're talking. And I like reaching out to the idea of, well, I don't know if I like the idea of reaching out to Elon Musk, but he's kind of a strange character. But if you wanted to fund it, I suppose I could keep him at arm's length. Have fun with the species description. I like that. And I like that was one of the things that I was hoping you would take away, that you might take away from the the description assignment, is the fact that there's kind of an element of joy in that description and trying to share your discovery with other people, and then who you're going to name it for. Address those species that were described before 1900 to see if those descriptions are still accurate. Yeah, and that would be ideal, but it's really hard to do when we don't know, unless there's good geographic coordinates, for example, really explicit to say we collected it here, and then you could go back to there, collect things again, and sequence, and maybe determine, kind of infer which ones those names we're talking to. Now, when you say based off new DNA data, are you hoping to describe things just based on DNA? Because some people do that. There's some groups where, with fungal description, most fungal descriptions, most bacterial descriptions now are based strictly on DNA. Should we be doing that with eukaryotes? Okay. As we keep discussing this, maybe what I'll do is I will try to open up a poll where I'm going to ask you specifically about the role that DNA plays in species descriptions. How important, what role should DNA play in species descriptions? And give me your thoughts there. And then so use the DNA, I guess, to help aid you, but not strictly based off of DNA, though. And then the suggestion that we should also base eukaryotic descriptions on morphology, morphological features. So how do you deal, if I can keep playing devil's advocate, how do you deal, with cases where there aren't a lot of morphological features, where, as I said in the bullet there, they're so morphologically depauperate that we don't have things to hang our descriptions on. 90%. All right. So it seems like we are, let me share that, stuck or captured in two different groups, kind of on a 3-to-1, or 3/4 of you believe critically important, all description should be urged to contain DNA, and somewhat important, use if convenient. Maybe before we leave this particular exercise, what advantages might DNA have compared to morphological features? What might there be some what are some of the advantages to including DNA in your description, rather than just morphology? I think DNA should be used, but not the be-all and end-all, as there can be huge similarities in very different species. This is true with DNA, that there's no kind of magic divergence threshold beyond which things are always the same. But actually, the same is then true that you can have morphological crypsis with deep genetic divergence. Morphological features can be more generalized. DNA can give us clear evidence. You would see in the tree that you showed us that many species with the same name were actually shown with DNA to be different species. DNA can be more accurate since many species have the same features but may not be closely related for the Nematerens. These are all really good thoughts. I'm going to give you one more to think about, and it's going to kind of think about some of the groups that we're going to come to next week. for example, with spiders. Taxonomists tend to have, there's biases in all parts of science. Those historical biases are often sex-based. And most taxonomists are male. Most species descriptions that have been completed by male taxonomists have been done on male samples. And so for most groups of invertebrates in the world, if you catch a female or an immature individual, even if there is a taxonomic key or a species description based on an adult, it's likely based on the male, and it's unlikely that you're going to be able to identify it based on morphology alone if what you've captured is a female. With some departures, most things, the departures from a 1-to-1 sex ratio aren't that common. So on average, most of what you're going to be catching for most invertebrate groups is not going to be identifiable because half of the adults will be female and not identifiable with the key that you've got, and then the vast majority of them will be young and so not identifiable with the key that you've got. And this, in fact, to me is one of the main pluses to incorporating DNA into species descriptions, because the DNA aspect is irrespective of the sex or of the life history stage. And so if your description incorporates DNA, standardized DNA like a barcode or other regions, that gives other researchers the capacity to, even if your description is based only on males, it gives the world the capacity to extend your description to include the female once perhaps we capture it and understand what it looks like. It also, for many invertebrates, gives us the possibility of capturing larval stages. We talked about trogophore and villager and all of the Pylidium larvae, all these different kind of larval stages. Oftentimes, we have no idea how those actually align and who belongs to whom, and we'll come to some of that later on. And I'm glad I've prompted a discussion in the chat here. This is great. Well done, you. Hands up, pat yourself on the head, and we will keep going. And I'm not going to vérifier l'achat because I am right here, right now. But we're going to keep going and say that the likelihood that we're going to get through 3 different groups today is slim, so there'll probably be a nematophyren. These are the best things about nematophyren's later on, posted asynchronously. But the next group that we're going to get to, these other thread-like things, in this case, the Nematodia. So we've left the spiralian group behind, and here in the Dun phylogeny, we've jumped over to a clade, a well-supported clade called the Nematodia that contains nematodes or roundworms and nematophyres or hairworms. These, of course, and so you can see a real change, couple of order of magnitude difference in the number of described species. Nematodes are amongst, pardon me, my use of the mouse there. Stop, Doug. Amongst the most diverse groups. And so we'll start out with the nematodes. And I love this quote. This is a nearly more than 100-year-old quote from Nathan Cobb, the father of American nematology, that if all matter in the universe except nematodes were swept away, the world would still be dimly recognizable. So if you want to transport that to 100 years later, with no spoiler alerts, I think necessary, but in the Marvel universe, in Endgame, as people begin to dissolve away and disappear, they should have been a small brief shadow of nematodes left behind as all of those characters disappeared. I hope there's no spoiler alert or spoilers given there. So these are uniquely smelling and tasting kind of things. So they have a complete digestive system with a bunch of different cephalic organs called amphids and phasmids so amphids on the front and phasmids on the back, that are all based on smelling and tasting. And they become real kind of a characteristic group for some of what I've been advocating for when you've heard me talk about in the past, not how much of a vertebrate bias and a primate bias it is for us to use the word I see to mean I understand. Because there's so many things that we don't see, so many different wavelengths that we're not receptive to at all, that some of the other species that we share the planet with are. So the way that we see is not the way that the world sees at all, let alone the fact that most species on the planet don't care at all for seeing. Seeing is not understanding. To understand To empathize means to taste and to smell, and that's the nematodes. They're not really visual. They might have some negative phototaxis here and there, but these are smellers and tasters. And I'm going to talk about one particular example later on where we've used that smelling and tasting to our own medical benefit. So no special circulatory or respiratory systems. They tend to be very small. Interestingly, no circular muscles here. So they use only longitudinal muscles, and we'll talk about how they move. They're gonochoric, and you can tell males with their hooked posterior end, and they live everywhere. Marine, freshwater, terrestrial, free-living, and parasitic, and in endgame, no. In the endgame, there'd be a dim shadow left behind of nematodes. What about the Spider-Man, though? Well, what do you mean, in the multiverse, or the fact that it was off-world? Because even if it's off-world, we still carry these things with us. And I'll give you an example of that if you were a manatee, because wouldn't we all like to be manatees on some days? There's no way I'm getting to the Nematophyrens today. I'm having too much fun. So like a lot of the ecdysozoans, which I'll break down, and you've already talked about in lab, so you know this. This is the group, this is the clade that's described basically by molting, by a complex layer of cuticle, three layers of cuticle. But in this particular cross-section, what we're looking at here are the longitudinal muscles, big fibers of longitudinal muscles going from anterior to posterior. They have a hemocoel that's reduced in free-living, that's a nervous system that's entirely within the epidermis, and no circular muscles at all. And so the hemocoel there in the epidermis has been exaggerated in the drawing for clarity because it is actually quite reduced. Doug, get out of there. My office mate is moving around. So, efficient locomotion for nematodes involves this kind of undulation, and it needs some kind of substratum or something for it to bounce off against, because otherwise it just becomes... That's it. You ding-dong. I was down in the basement there with the dog for a second. They need something to bounce off against. So you can see in this video on the left-hand side as it's scrolling to how dirty, how much substrate there is on that cover slip. And so there is this undulating anguilliform or eel-like motion is possible in a dorsal ventral plane rather than side to side like eel. So it's kind of like that. But then when it's a much clearer slide over here, if I restart that video, you can see if they've got nothing to bump into, they're just flailing back and forth. The dancing nematodes, perhaps. So from your text, you can see in Chapter 18, where we are for the Nematoidea, you can see that there are stretched and contracted longitudinal muscles running along the body and firing kind of on the inside of each curve, that when there is a substratum or something viscous or an obstruction with which the body can bounce off of, they can begin to kind of move their way forward. So, a lot of, like I said, these are smellers and tasters. So some amazing adaptations. So up at the front end of the body, the amphids, and that they have mechano and chemoreceptors. So phasmids near the tail end and amphids near the anterior end. You can see a lot of these, and in the mouth, right in the center of this diagram here, lots of different sensory papilla, cephalic, outer labial papilla, inner labial papilla, and these amphids. All of these things are about sensing, tasting, responding to vibration, responding to different chemical signals in the environment. These are the smellers and tasters. So, as I said, they're pretty much any way that there is to live, these take advantage. They can be carnivorous, they can be herbivorous, they can be bacteriivores, fungivores, deposit feeders, they can be parasitic, and a lot of how we identify them taxonomically has to do with differences in the stylet, which you'll see here, and the, what did DVL, dorsal, ventral, and lateral, I believe, is what DVL meant on the nervous system slide one ago. The muscular pharynx, and so these are about kind of, the stylet is about piercing. If, for example, if it's going to be eating a plant cell, piercing the plant cell and then using that muscular pharynx to suck out different materials. Now, if they're parasitic, if they're living in terms of parasitically in terms of inside of animals or plants, or if they're carnivorous, eating mobile things, there's real adaptations around the mouth into not just these sensory labia and papilla, but also into teeth. So teeth to make sure that if you're going to eat a little writhing protozoan Ciliopheren or something, back from way, way, way earlier in this year when we talked about protozoans, the teeth are then kind of curved inwards so that it's much harder for that prey item to get out, and then much less complicated but more intricate kind of teeth associated with maybe grabbing onto a particular part of the digestive system of whatever it is that you are the internal parasite of. So the muscular pharynx that I talked about, this is the triradiate muscular pharynx. So you can see here that there are these 3 branches of cuticle that I'm tracing on the interior of the slide. So that's that's an adaptation, increase in surface area that's surrounded by muscles that is increased suction capacity. And the pharynx is that pump that sucks the food out into the intestine. And then there's 3 bands of muscle that fire in order to contract and expand the lumen. No nephridia, no true nephridia. These Rennet cells, these are, you see here, these kind of large hanging tongues beside the intestine. These are likely doing the job for osmoregulation. They're excreting and where they're concentrating excretory products into the intestine. There's a whole bunch that physiologically, just for all 3 of these groups that we're talking about today, that we have yet to understand. But so the question mark beside secretion is that they perhaps also produce the gelatinous matrix that tends to surround eggs when eggs are the glycoprotein that surrounds and protects eggs when they're ejected into the outer environment. But they do tend to excrete ammonia, and that is either sent out through the intestine or just diffuses across the body wall. Remember, we're talking about fairly small organisms here. Some hermaphrodites, but mostly gonochoric, where there can be internal fertilization and copulation. So we see females and males here where there is a seminal receptacle and then a spicule. The spicule, this is the hook that I was talking about, where I said that males can tend to be known by their hooked posterior. There's the sperm duct, and the sperm is aflagellate, so it's kind of it's amoeboid. It creeps along, it doesn't move, and not a fast swimmer here. But you can see the apparatus associated inside of this particular nematode with the sperm duct preparing and holding these amoeboid spermocytes before they're ejected into the female. So I mentioned one of the things that I wanted to talk about when talking about the nematodes today was the fact that as a smeller and taster, there's ways that we have used them scientifically, use that smelling and tasting to our capacity. This is a paper from a couple of years ago now, but it's called a nematode scent detection test. And this was the kind of, to me, really elegant and very transferable technology where C. elegans nematodes are attracted to urine from cancer patients while they shy away from non-cancer patient urine. And so the distribution of this is basically that this was a test that had a markedly higher accuracy rate than any of the other and more expensive and perhaps more prohibitive tumor markers that they would have to use. So this is a way that we may actually use some of these nematodes to facilitate cancer diagnosis. Now, of course, they can be parasites, so before we leave talking about question in the chat, what are the benefit of having such well-developed senses for the nematodes? And it's the fact that they don't interact with the visual world really at all. And so their capacity to track down whether, you remember all that list of life histories that they take advantage, that they're bacteriivores, fungivores, carnivores, herbivores, free-living, parasitic, all of that has to do, many of those strategies have to do with tracking down a prey item and being able to identify it. And while there are some generalists, many nematode species are actually more specialized. And so not just tracking down any particular food source, but one particular species in particular in many cases. And so that requires you to smell and taste with some degree of accuracy your way around the world if you're going to bump into the thing upon which you need to feed. So lots of different parasitic forms, and so we're looking at some pig intestines with some parasitic forms. So the pig intestine is the large sleeve there with the nematodes bursting out of it. So these are Ascaris worms on the bottom, and the spaghetti, which always looks like spaghetti to me until I read the bullet, extracted from the intestines of a single child. What a bad day that would be. So the eggs, and remember we talked about the positive use of DNA in terms of species descriptions to associate different stages of life history. Many different eggs and are then also like larval stages where we don't know whose eggs are associated with whom when we find them in the soil. But some eggs of these Ascaris worms can last for 20 years outside. This is a video. I'm not going to show the whole thing. I'm just going to jump through it. You can see it's 7 to 8 minutes long. This is a video produced by the vet college here. And Andrew Peregrine. Dr. Peregrine is talking about a giant kidney worm that was found in the urine. So eggs in this case were found in the urine of a dog. Rosie was the dog of a student who took this course in 2015. And this is a video of the operation. And you can see the size there of the kidney worms that were taken. So while some of them are very small, some of them can actually, some nematodes can be quite big. So these are, gorgeous. But I'm sure Rosie is very happy to have them out of her. And there she is, happy that the operation is over. Yay, veterinary care. Nematodes can also be an incredibly economically damaging and sociologically damaging parasite of humans. So here's a loa in the eye of a human. It's a dracunculoid filarial nematode that can cause blindness, and it's transmitted by insects. And then, of course, you might have another dracunculoid, so another parasitic nematode called the Guinea Worm, where the definitive host, sexual reproduction, occurs in humans. Intermediate hosts are copepod crustaceans. The female worm can be up to a meter, close to 1.5 meters. And so what you're seeing here is the use of a matchstick. So breaking the worm, kind of respiring, having an opening through the skin. And what the physician or what the doctor is trying to do is to essentially wind up with a little turn of that matchstick every day and remove eventually the dracunculoid nematode. So 1.5 meters maybe of winding on the circumference of a matchstick. And there's some thought that this apparatus or this medical technology is the root for the catechus, meaning this symbol to essentially represent the medical profession. This is not about staphs and snakes, as some people think looking at that form, but it's in fact about dracunculoid nematodes and using matchsticks or small sticks to move them out, encourage them to leave their definitive human hosts. So in this case, what we're looking at in a lot of different parts of the world are infestations and affecting of human populations where there is no access to clean water source or to clean water source. And my body, my brain wants to encourage you to think, if some of you are naively thinking that, so that's not Canada, remember that most of the First Nation populations across Canada don't have access to clean water sources. So this is a problem for Canada and Canadians as well. So the human drinks unfiltered water containing the copepods here that contain larval stages of the nematode. When they're consumed, they move into the host stomach where they penetrate with styles and stylets, where they then mature and reproduce. The fertilized worm migrates to the surface of the skin, causes a blister, discharges larvae. Those are released into the water, and then those larval stages are consumed by the copepod, and the cycle continues. There's a lot of different diseases, human diseases, that are caused by nematodes. This is elephantiasis. This is a horrific one, transmitted by mosquitoes, but the adult worm lives in the human lymphatic system and then blocks the flow of lymph, leading to these kind of chronic and ultimately horrifically painful edemas. So this is looking at the life stage as well. So blood meals in this case for mosquitoes and shared. But again, so if there's blood meals, remember when we're thinking about mosquitoes, the adult mosquitoes, we're thinking about shared blood meals, but that also has to do with access to clean water sources, because where are the larval mosquitoes growing up? In water sources adjacent to towns. And then, of course, heartworm. So everyone who has a dog, so Doug, look, this is the reason that you're going to get medicated for most of the year. So this is transmitted by mosquitoes, but this nematode can be lethal to dogs, and so you're looking at a really unfortunate canine heart there, filled up with dirofilaria, which is the nematode heartworm. And he's a collie, Doug is, so he's going to be getting fairly specific medication, because his blood-brain barrier is just very different and sensitive. One last word about the nematodes, and this is a study that you might have seen because it was in the New York Times. This was an amazing study where in Florida, people were doing some interesting things and helpful things instead of just normal Floridian interesting but not helpful things. This was the discovery that in that kind of dense and gorgeous skin, the dorsal skin of the manatee, there may be upwards of more than 500,000 individual nematodes. Reconcile this with Cobb's earlier quote about the fact that if all of the universe was vaporized, there'd still be the dim view of all of the world. That's because of the shape of the nematodes. So you'd still see the shape if this manatee was in endgame and it vaporized. You'd still see the shape of the manatee because it has nearly more than 500,000 individual nematodes. Now that's not species, those are individuals on that body living in its skin, principally. But within this particular study, one of the other neat things that they discovered as they counted these nematodes was the fact that there were 3 probable new species discovered just in these half a dozen manatees that they explored in Florida. It's amazing to me. I'm looking at the chat right now. I see we have a couple of minutes left. I'm going to start off with the Nematophyrens for a couple of minutes because I can show you some amazing videos, and then I'll probably finish with a 5 or 10 minute asynchronous bit that you can watch at your leisure later on. So these Nematophyrens are a much less diverse group, but we know much less about them, and that's because they live, their life histories intersect with species that tend not to affect us. But we see them, so they're also triploblast. I'll show this again in the, there's a lot of these, if you go through the synapomorphies that characterize the Nematophyrens, there's a lot of these things, these thread-like organisms that are shared with the nematodes, or similar to the nematodes. They have very reduced but not absent circular muscles, and they look, there's not a lot to look at here. But so practically featureless, lacks a distinct head, coil together in masses that resemble a Gordian knot. So what was the Gordian knot? This was a famous kind of historical challenge to how to untie this knot. What wisdom could you show me by untying this knot? Well, the best way, as shown in this famous painting, is to do it with a sword. It's kind of like Indiana Jones when he's in the first movie where he's challenged to the sword fight, and he pulls out the gun and shoots. So that was his response to the Gordian knot. And we actually see the Gordian knot in the Batman and Superman Dawn of Justice film.
It's the sword of Alexander.
So this is purported to be the sword that Diana is looking at that cleaved the Gordian knot.
Enjoy.
Thank you.
It's a fake.
The real one was sold in '98 on the black market. Now it
All right,
hangs.
you know the film. Anyhow, the Gordian Knot appears in the Superman film and Superman versus Batman film. All right. So one of the neat things here is this proboscis. And so you see a very reduced internal organism. It's like I said, it's very small, slender. But the proboscis, I wanted to take, this is a video of the proboscis of a Nematophyren that's interested in penetrating insects. So the definitive host will tend to be insects. Insects contain writhing mass, so they live terrestrially. At some point, the Nematophyren controls the insect, takes it to water. When it bursts out of their body, produces eggs that turn into the larva that then jump into a secondary host and then are consumed by the terrestrial definitive host. So most of the stages of their host life cycle, we don't actually know for most species, but this is what it looks like when they burst out of the body. This is an unfortunate cricket that has been mind controlled by that Nematophyren, brought to water, jumps, and then the Nematophyren jumps out of it. That's where I'm going to stop today. I'm going to pause the Zoom land. I'm going to be around for the next hour or so if there's particular questions. These are the digital office hours, so it gives me, Doug and I, a chance to get some water and refill the tea, and then if anyone's still around has some questions, I can answer those as I prepare materials to go up online. Other than that, have a great weekend, and it's going to be gorgeous weather. Please get outside and do something good for your body and your brain. I think highs of 13 degrees Celsius and sunny both Saturday and Sunday. So enjoy the weekend, and thank you. Stop the share.